Hi, this is Mr. Laudermouth. Good morning, good afternoon, or good evening to you, as the case may be. I want to give a brief talk today in this video on an aspect of photosynthesis, the Calvin cycle specifically, and how evolution has led to different adaptations in different environments, allowing plants to deal with a problem that crops up when they're trying to successfully carry out the Calvin cycle. So we're going to talk about some variations on a theme. Let's start by just reminding ourselves quickly about what plants need to successfully do photosynthesis. The light-dependent reactions, of course, need sunlight from the sun, and they need water, which they take in through their roots from the soil. The Calvin cycle, the light-independent reactions, need carbon dioxide, which comes from the air. Now, what structures have plants evolved to supply these needs? Well, of course, they've got roots with root hairs to increase maximum absorption of water and minerals, and they've evolved leaves as sites both for chlorophyll to capture sunlight and to process the carbon dioxide to make sugars. So let's focus on the leaf a little more in detail. It can be divided up into a couple different layers, like the palisades layer, the spongy layer. There's an epidermal layer, which, just like our epidermis, our outer layer of skin, is protective. It's covered by a waxy layer called the cuticle that helps trap water inside. On the underside of the leaf, we'll find openings called stomates or stomata. Those openings are surrounded by a pair of cells called guard cells. They basically guard the opening. They guard the stomata. They allow it to either be open or to be closed. When it's open, water can leave out of the leaf through the stomata through the process of transpiration that helps plants move water up from the roots to the leaves without spending energy, so that's a good thing. It allows the oxygen that builds up in the leaf as photosynthesis occurs to exit the leaf. That's a waste product from the standpoint of photosynthesis, so it's good that can leave the plant. And of course, it's allowing carbon dioxide from the air to diffuse into the leaf. And the Calvin cycle in the chloroplast requires that carbon dioxide in order to make sugars. So having those stomata open all sounds like a really great thing. It's allowing for gas exchange. The water will end up passing into tissue called xylem. The sugar gets moved through a tissue called phloem. We'll look more at that in a future unit. But it's not all as easy as it looks. We have to remember that plants can have a limited supply of water, especially if they live in dry environments. So on hot or dry days, plants will quite naturally close their stomata to conserve water. If a plant runs out of water, it's going to wilt, it's going to lose turgor pressure, and with no water, there's no electrons, of course, to run the electron transport chain, and you can't do any of the steps of photosynthesis. So those guard cells, they will actually lose water through a process of transporting out ions, and then the water flows out of the guard cells by osmosis, allowing them to close, and that process can be reversed to open up the guard cells and open the stomata. So this is a good adaptation if you live on land in an environment where water is not always in constant supply. But just closing your stomata whenever it gets too hot leads to other problems. So this is under normal conditions. When the stomata is open, water can leave, carbon dioxide can come in, oxygen can leave. But as soon as I close those stomata, yes, I'll trap the water inside, which is good for the plant, but I cease all gas exchange. No more carbon dioxide is going to come in, so I really can't do the Calvin cycle, but I can still do the light-dependent reactions because all that requires is sunlight and the water, which I'm trapping inside. The water is going to be split to release the electrons, but that's going to produce oxygen gas. The concentration of oxygen is going to start to build up inside the leaf at the same time that we're running out of carbon dioxide. That's going to cause a very serious problem for the Calvin cycle. Best laid schemes. Where does this problem stem from? Well, if you recall, the main enzyme involved in the Calvin cycle is called rubisco for short. It's got a longer name. It's the enzyme that fixes carbon. It takes the carbon from carbon dioxide and it bonds it to a 5-carbon molecule called RUBP. creating an intermediate molecule that gets broken down in a couple steps and ultimately will produce carbon molecules that can be used to synthesize sugars, glucose. Carbon dioxide is the optimal substrate for Rubisco to use in the process of the Calvin cycle. So it will reduce RUBP by adding the carbon. This will, again, in the end, result in us building sugars. All good. That's what we want from photosynthesis. But if we close the stomata, as we run out of carbon dioxide and the amount of oxygen in the leaf overtakes the amount of carbon dioxide, oxygen will actually become a competitor for CO2 in the active site of Rubisco. It basically becomes a competitive inhibitor. But it actually goes even beyond that. Not only does it inhibit the Calvin cycle, Rubisco can use oxygen along with RUBP and catalyze a reaction. It will bond O's to RUBP. That, though, is not going to result in us building any glucoses. Instead, we're going to oxidize RUBP. We're going to break it down into smaller pieces, not build up sugars. We're basically going to be doing the opposite of what we want to do in photosynthesis. This process that breaks down sugars is called photorespiration. From the plant standpoint, this is really, really bad. So again, here's we're looking at the normal Calvin cycle when we have plenty of carbon dioxide. Rubisco will join a carbon to the 5-carbon RUBP, and by the time we're done, one complete turn of the Calvin cycle, we'll have made some G3P, which we can combine to make glucose sugar. And the plant's very happy. This is the normal Calvin cycle, or what we call the C3 cycle. Most plants in most environments just do this normal cycle of C3. But these plants, again, if they have to close their stomata to save water, there's a danger that the oxygen level gets too high. Rubisco will combine RUBP with the oxygen, and all we'll end up doing is breaking apart RUBP. And along the way, instead of making sugars, we'll actually lose more carbons. We'll lose 2 carbons along the way as carbon dioxides and not make any ATPs in the process, not produce any energy. So not only are we running out of the starting materials to do the Calvin cycle, we're running out of the ATPs we actually need to power the Calvin cycle. So we're losing both basically substrates and the energy to run the process. That's why photorespiration is pretty bad for the plant. So we're short-circuiting the Calvin cycle. We're losing carbons that could otherwise have been used to make a sugar. We're losing them as carbon dioxides. Up to 50% of the available carbons can be lost this way because of photorespiration. So we're not producing any ATPs. We're not producing any food. We're not producing any sugars. If we could just find a way to limit photorespiration, we could make the Calvin cycle up to 50% more efficient. Well, that's the kind of advantage that natural selection would definitely pick out if any plants evolved that kind of adaptation. If they had a different way of fixing carbon, avoiding the photorespiration problem, they would have a definite advantage over other competitive plants in their environment. Well, what I'm getting at, as you probably guessed, is that some plants have in fact evolved mechanisms to get around the photorespiration problem. They have found different ways to fix carbon so that they can complete the Calvin cycle more effectively. The 2 groups of plants best known for these adaptive advantages are called the C4 plants. They separate the carbon fixation step of the Calvin cycle physically. They carry it out in a different group of cells away from the other steps of the Calvin cycle. That way, basically, they're not allowing oxygen to interfere with the process. They will store that carbon and then use it in other cells in the leaf. To get around the problem of Rubisco bonding to oxygen, they fix their carbon with an entirely different enzyme. They use an enzyme called PEP carboxylase, which does not bond very readily with oxygen, but it still will capture the carbon dioxide. The other group of plants who have evolved a different mechanism for doing the Calvin cycle are called the CAM plants. They separate the carbon fixation step and do it at a different time of day from the other steps in the Calvin cycle. They mainly fix their carbon dioxide at night. They again store the carbon for later use in a 4-carbon compound. Generally, they'll store that carbon overnight and then do the rest of the Calvin cycle during the day. We'll talk in a minute about why that's an advantage. Let's start with the C4 plants. Corn and sugarcane, important crops that we grow, are C4 plants. Both of them originate from tropical environments, even though corn today we tend to think of as growing more like in the Midwest here in Illinois. It originated in central Mexico, a much more tropical climate. Sugarcane still grown in the tropics. The first step in the C4 pathway for doing the Calvin cycle is to take the carbon dioxide through your stomata and store it as a 4-carbon compound using that enzyme PEP carboxylase. Now, PEP carboxylase is definitely adapted to hotter, drier climates. Even if the stomata close, the PEP carboxylase will not use oxygen to do this carbon fixation step. It will only do it with carbon dioxide in the leaf. The advantage of doing this: if you fix the carbon in an outer layer of cells called the mesophyll, store it as this 4-carbon compound, oxaloacetate, which is turned into malate, you can then shuttle that 4-carbon malate into an inner layer of the leaf called the bundle sheath. This is getting farther away from the stomata, so farther away from all the oxygen. That bundle sheath inside there, it's a much more protected environment. Malate will break down, release the carbon dioxide. That carbon dioxide can then go into the normal Calvin cycle. It's fine to use Rubisco in the bundle sheath because the concentration of oxygen there is much, much lower. We're away from the outer layer of cells where most of the water splitting and the oxygen production is occurring, so there's much less danger in the bundle sheath of the Rubisco bonding to oxygen instead of CO2. So again, all the gas exchange is happening in that mesophyll, that outer layer. We'll store the carbon dioxide in a 4-carbon compound, move it into the bundle sheath where there's much less oxygen, release that CO2, Rubisco will grab onto it, use it in a normal C3 Calvin cycle, and we'll make our sugars. And we've avoided, by and large, the danger of combining the carbon compounds with oxygen that will happen in photorespiration. So again, PEP carboxylase, much better to use to fix carbon than Rubisco. It has a much higher attraction for CO2. Then we'll release that CO2 in the inner bundle sheaths away from the oxygen. So if we compare C3 to C4 plants, you can kind of see here that that upper layer, that mesophyll layer, is much bigger in C3, much smaller in C4, and the bundle sheath is much more extensive in the C4 plants. So we're separating kind of Calvin cycle into 2 different stages, into 2 different layers of the leaf. CAM plants, CAM is short for crassulation acid metabolism. These are also plants adapted to hot, very dry climates. Many desert plants are CAM plants. They do their carbon fixation at a different time than the rest of the Calvin cycle. They can keep their stomata closed during the day and save their water. Then if they open their stomata at night, it's much, much cooler, much less chance of water being lost through the leaf, so we're not losing much water. The stomata can be open at night and the carbon dioxide can come in. Again, we'll store that carbon dioxide in a 4-carbon compound. In the day, as the temperature increases, the stomata can close to save the water. Then the 4-carbon compounds can be broken down. We'll release the CO2 and we'll use it in the normal Calvin cycle. So again, here we're avoiding the danger of having our stomata open all the time and losing water during the day. And by storing it in a 4-carbon compound and then using it during the day, we're largely avoiding the problem of photorespiration. Succulent plants like cacti, but pineapple as well, are CAM plants. Again, some more examples here. So just to do a quick summary, in both cases we have plants having to deal with the issue of both needing to exchange gases, carbon dioxide for oxygen, and avoid the problem of excessive water loss. C4 plants separate the steps of the Calvin cycle into different cell layers. They physically separate it. They fix carbon in one cell layer and then release the carbon into another cell layer where the Calvin cycle can occur away from oxygen. CAM plants take in their carbon dioxide at night when it's safe to open the stomata. You won't lose much water. And then they'll run the Calvin cycle during the day with the stomata closed to avoid losing all the water at the hottest part of the day. So separation anatomically versus separation temporally, both really to overcome the same problem of photorespiration and water loss. Why should any plants still use the C3 pathway if it carries this danger with it of doing photorespiration and losing ATPs and carbons? It's thought that Rubisco originally evolved at a time when the atmosphere on Earth had a much higher concentration of CO2. So for plants living on land, photorespiration probably was not much of an issue because there was always lots of carbon dioxide around. Today, though, CO2 levels in the atmosphere are much lower than they have been in the past. So now that there's 21% oxygen in the atmosphere and just 0.03% carbon dioxide, photorespiration can be a serious problem. On a hot day, a plant doing C3 can lose up to 50% of its carbon by photorespiration. That's a strong selective pressure to evolve a better way to fix carbon and avoid the problems of photorespiration. I hope you have found this helpful, and I'll see you in class.